So now that we've talked about lasers and how to make a laser, very bare bones version of it, and why they're so great, let's talk about some of the techniques where lasers can be used in chemistry to do spectroscopy. So because of some of the advantages, we can do some pretty unique experiments, and we're only going to touch, the very basics of laser spectroscopy and types of spectroscopy in this. I'd be happy to talk to you more about laser spectroscopy and the different sorts of techniques that are used if you are interested. So the simplest way we can take advantage of lasers in spectroscopy is doing a type of experiment known as direct absorption spectroscopy. And the idea here is that we basically just replace a lamp with a laser and doing traditional spectroscopy experiment. So we have a tunable laser, some laser where we can change the frequency of it, send it through our sample, and then we have a detector to observe the light. So very basic experiment. The reason this is helpful is that because we're using a laser, we can take advantage of the fact that the laser is monochromatic, and that lets us get light of a very specific color, and this lets us do high resolution experiments. And so I've taken advantage of this in my research. So I use a tunable laser and do direct absorption spectroscopy. The other thing is that it's very bright. So we get the advantage of having lots of light, and that can improve the signal to noise ratio on our experiment. Kind of the next step you can take here, and which is what we do in our group, is we can add what's known as multipass, a multipass cell on top of this. And so the difference here is that instead of our sample just being in some cell, we have it in a special cell. So our sample's in the middle here. And we take advantage of the directionality of a laser to send the light multiple times through our sample, using the the optics of a specific set of mirrors at a specific angle and things like that. So we get longer path length through our sample, which leads to a larger signal and a more sensitive measurement. So this lets us measure things at very low concentrations. For example, in my lab, we measure isoprene using multipass direct absorption spectroscopy, and we can measure concentrations down into the parts per billion level. So, where we have tens of isoprene molecules for every billion other molecules that are present in our sample. And so these are the sort of concentrations we need to detect isoprene in its natural environment in human breath and the atmosphere. So this is a nice technique for doing that. Now you can take that one step further and do a technique that I used in graduate school, which is known as cavity ringdown spectroscopy. And this essentially is a similar idea, except that instead of our having two specially designed mirrors and we get a few passes, we have essentially almost like another laser cavity where we get 99% plus reflective mirrors. And instead of just a few passes, we get thousands of passes. So in a typical multipass cell, you maybe get a few hundred passes, whereas cavity ringdown, you get thousands of passes. And so you can get path lengths up to kilometers in something that can fit on a regular optic table. And so cavity ringdown spectroscopy only works with lasers. You can't do it with any sort of lamp. And the advantage of this is that you can go to even lower concentrations. So you can get very low concentrations with this. And this relies, I'll just note that the cavity that's part of cavity ringdown is this set of mirrors. Your sample is in the middle here. And, these are very highly reflective mirrors. They cost a lot of money. I know this from personal experience. And can be very tricky to align. But the one key thing is that the fact that the laser is monochromatic actually matters for this experiment, because the cavity can only support certain colors of light at a given time. And so we take advantage of that fact, and our laser has to match up with our what's called a ringdown cavity to be able to do this measurement. This is great for things like transient molecules, or van der Waals clusters that you can only make in low concentrations. And so you can measure these things like this, with a technique like this. So, for example, one of the molecules I studied in graduate school was the cluster of two D2O heavy water molecules stuck to each other through hydrogen bonding, or I guess deuterium bonding in this case. And so we could actually measure that in the gas phase. And that's, we needed cavity ringdown to have sufficient sensitivity to be able to measure the few, D2O molecules that are clustered out of, a bunch of other stuff that was present in our sample. So these are just a few. This is just a very, very small flavor of the types of things you can do with a laser. And in the next video, we'll talk about using the fact that you can also generate laser pulses that are very short to do time resolved spectroscopy.